Selamat siang dan selamat datang kepada seluruh peserta yang berpartisipasi pada webinar Automation Solution, Services and Data Integrity for Pharmaceutical Laboratories. Perkenalkan, saya Dewi yang akan menjadi MC pada webinar hari ini. Sebelum kami memulai webinar ini, kami ingin menginformasikan bahwa mikrofon dan video peserta telah kami nonaktifkan. Jika Anda memiliki pertanyaan untuk para pembicara, silakan kirimkan pertanyaan Anda melalui Q&A box yang tertera pada layar Anda. Jika terdapat gangguan pada internet selama webinar berlangsung, mohon menunggu dan silakan masuk kembali ke link webinar. Sebelum memulai webinar, izinkan saya membacakan agenda acara siang hari ini. Welcoming oleh MC, kata sambutan oleh Ibu Liliana Budiharjo selaku Direktur PT Gaia Science Indonesia. Pembukaan oleh moderator oleh Bapak Simon selaku Sotax Product Manager PT Gaia Science Indonesia. Presentasi pertama oleh Mr. Jürgen Kempf selaku Sotax Distributor Manager for Asia Pacific dengan topik Automation Solution and Data Integrity. Sesi tanya jawab, kemudian presentasi kedua oleh Mr. Jean-Louis Raton selaku SPS Business Development Manager dengan topik SPS Pharma Services. Kemudian sesi tanya jawab, kalimat penutup oleh MC, serta penutupan. Berikutnya kami akan putarkan video pameran Lab Indonesia 2018.
I'm very surprised. There's lots of people coming, and many people is interested in Japanese products. So I'm very happy to see that.
very fantastic event, and like I said, it's exceeded my expectation. I'm very happy with Lab Indonesia.
I think it's pretty good. You should just continue doing that because the application of the analytical chemistry is quite important. I mean, it can expand to a different discipline. So yeah, just keep doing it. We are going to do it.
Demikian video singkat Lab Indonesia. Selanjutnya adalah kata sambutan yang akan disampaikan oleh Ibu Liliana Diharjo selaku Direktur PT Gaia Science Indonesia. Kepada Ibu Liliana untuk waktu dan tempat, saya persilakan.
Pelanggan yang kami hormati, selamat siang dan salam sejahtera untuk kita semua. Saya mengucapkan terima kasih untuk Bapak dan Ibu yang telah meluangkan waktu yang berharga untuk mengikuti webinar laptop series kami dengan topik Automation, Services, dan Data Integrity for Pharmaceutical Laboratories. Dalam kesempatan yang baik ini, PT Gaia Science Indonesia selaku distributor tunggal dari Sotax, dengan total pengalaman kami bekerja sama dengan Sotax selama 20 tahun, kami mengundang Mr. Jean-Louis Raton dan Mr. Jürgen Kemp sebagai pembicara webinar yang membawakan topik-topik hangat yang saat ini kita semua membutuhkannya. Kami juga berterima kasih kepada Bapak dan Ibu yang telah menjadi pemakai setia Sotax dan mendukung Sotax menjadi satu brand yang dikenal bermutu tinggi sebagai alat pengujian produk-produk Pharma. Dalam kesempatan kali ini, topik yang kami angkat adalah automation. Topik ini kami angkat sehubungan dengan masa pandemi. Masa pandemi ini memberikan kita semua tuh banyak kejutan yang mendadak ya. Nah, automation ini sangat membantu keberlangsungan produksi ketika pandemi berlangsung. Ketika pemerintah memperlakukan aturan bekerja dari rumah, ketika penggunaan ruang dibatasi hanya 50% daripada kapasitas maksimum. Nah, dengan automation ini produksi bisa dipertahankan untuk tetap normal dan stabil. Automation juga menjadi satu solusi terbukti di Eropa dan Amerika yang sudah banyak yang sudah menerapkan, dan hal ini juga menjadi tren baru di Asia. Semoga dengan pembahasan mengenai automation solution juga memberikan kita semua satu wacana yang kita semua akan pikirkan dan diskusikan. Mengenai topik data integrity, juga kami angkat sehubungan dengan audit ya. Hari ini sudah jadi makanan sehari-hari untuk masyarakat farmasi. Nah, audit ini juga menjadi satu tantangan bagi kita semua, bagaimana bisa mengatasi masalah ini dengan baik. Nah, kami berharap dengan pembahasan mengenai data integrity dapat memberikan solusi dan juga dapat mempersiapkan Bapak dan Ibu untuk kebutuhan di tahun-tahun mendatang. Dalam kesempatan ini kami juga memperkenalkan SPS Pharma. Jadi SPS Pharma ini adalah satu perusahaan yang menyediakan jasa untuk membuat metode analisa disolusi untuk produk-produk baru dari perusahaan Bapak dan Ibu. SPS Pharma sendiri berlokasi di Swiss. Nah, biasanya ketika produk baru itu akan dihasilkan, kita juga perlu merumuskan ya metode uji dan parameter-parameter yang sesuai untuk pengujian produk baru tersebut. Nah, SPS Pharma memiliki pengalaman bertahun-tahun, pengalamannya lengkap, juga didukung oleh peralatan yang lengkap. SPS Pharma bisa membantu kita semua untuk menghemat banyak waktu dan tenaga dalam membuat metode uji, terutama dalam parameter dan uji disolusi. Akhir kata dari kami, tak kenal maka tak sayang, maka kami akan menayangkan video singkat yang memperkenalkan Gaia Science secara keseluruhan sebelum kita memasuki sesi webinar hari ini. Gaia Science selalu memberikan upaya dan komitmen terbaik kami untuk menyediakan alat-alat yang bermutu tinggi, juga disertai dengan layanan instalasi, training, purna jual, dan kalibrasi untuk Bapak dan Ibu sekalian. Dengan ini saya mengucapkan salam sehat dan sukses selalu untuk kita semua.
Bekerja di industri farmasi, khususnya di laboratorium quality control, menuntut kecepatan dan standar GXP yang tinggi. Untuk itu, kita membutuhkan support agar kedua hal tersebut dapat tercapai dengan baik dan benar. Gaia Science memiliki tim engineer yang responsif dan antisipatif untuk memberikan solusi di lab terkait dissolution dan disintegration tadi. Good job, Gaia Science.
Bekerja selama 6 tahun lebih dengan Gaia Science, saya menyaksikan progres yang luar biasa. Bernaung di bawah perusahaan farmasi asing, kami dituntut untuk melakukan percepatan implementasi GXP. Dan Gaia Science memberikan respons yang luar biasa dan antisipatif. Untuk itu, sukses Gaia Science.
Terima kasih Bu Liliana atas sambutannya. Webinar hari ini disponsori oleh Sotax, SPS Pharma Service, dan PT Gaia Science Indonesia. Selanjutnya, webinar hari ini akan dipandu oleh Bapak Simon selaku Sotax Product Manager PT Gaia Science Indonesia sebagai moderator. Kepada Bapak Simon, untuk waktu dan tempat, saya persilakan.
Terima kasih Bu Dewi. Terima kasih untuk Bapak Ibu semua yang sudah hadir. Salam bahagia kepada Bapak Ibu dari keluarga besar perusahaan farmasi se-Indonesia. Salam bahagia, semoga Bapak Ibu semua dalam keadaan sehat dan mengikuti webinar ini sampai selesai. Kami mengangkat topik automation data integrity. Ini karena kami melihat kalau 10 tahun ke depan kemana dunia farmasi akan menuju, maka itu adalah menuju ke data integrity. Ya. Saya akan share presentasi. Semoga Bapak Ibu bisa melihat semuanya. Jadi untuk agenda hari ini tadi opening sudah selesai dilakukan, kemudian introduction oleh Ibu Lili selaku Direktur PT Gaia Science, dan sebelum masuk ke sesi automation, ini sekilas foto tim Gaia Science yang siap melayani lab farmasi, QC, R&D maupun IPC di perusahaan Bapak Ibu semua. Ini kantor kami di Kelapa Gading. Jika Bapak Ibu sering mampir ke Kelapa Gading, itu kantor kami ada di belakang Belatera. Ini seminar yang pernah kami lakukan beberapa tahun lalu sebelum pandemi. Sotax seminar juga, di mana 2 tahun ini kita tidak bisa melaksanakannya, sehingga kita membuat webinar ini. Ini adalah exhibition, jadi dari Lab Indonesia. Ini produk-produk Sotax dari Dissolution tester, physical tester, maupun automatic sample preparation. Jadi selain ada Dissolution tester, physical tester, sebenarnya Sotax juga ada automatic sample preparation yang selama ini di lab dilakukan secara manual. Jadi Dissolution sendiri ada yang manual sampling, semi-automatic, hingga fully automatic. Kalau fully automatic itu maksudnya Bapak Ibu nggak usah mencuci vessel, tapi dia akan mencuci sendiri. Nanti detailnya akan dijelaskan. Ini untuk disintegration tester automatic, manual detection, hardness tester dari yang manual, semi-automatic, hingga fully automatic, disintegration variability tester, tap density, dan captorcy tester. Jadi dengan QDOC adalah memberikan solusi separuh dari peralatan laboratorium yang ada di terutama QC dan IPC ya. Jadi ada 30 instrumen yang bisa di-connect dengan QDOC melalui drivernya masing-masing. Sehingga satu software untuk semua data begitu. Jika kita lihat dari workflow perusahaan farmasi, dari API, excipient, dari raw material sampai ke shipping produk, itu melalui beberapa tahap dispensing, mixing, granulation, drying, milling, mixing lagi, tablet press, coating, kemudian packaging, ya. Kemudian untuk data sendiri banyak sekali dari production data hingga ke packaging. Tentu untuk memperoleh quality data kita membutuhkan banyak aspek hingga setiap batch itu rilis dengan cepat ya, selain memenuhi syarat semuanya. Jadi di sini banyak melibatkan peralatan-peralatan, mesin-mesin dari yang manual, semi-otomatis maupun fully automatic. Jadi dengan memudahkan data, penanganan data, sehingga kita bisa lebih fokus untuk menangani hal-hal lainnya. Karena kalau kita membandingkan antara data yang diinput manual dengan setelah kita menerapkan data integrity, itu saya mengambil satu istilah di bahasa Jepang, itu beginning is easy, continuing is hard. Maksudnya untuk manual input memang kadang-kadang kita tersenyum dalam tangis. Artinya setelah melewati tahap itu, begitu kita kena audit, dengan banyaknya data-data yang harus kita siapkan secara manual, itu akan bikin kita pusing. Sedangkan kalau kita udah menerapkan data integrity, memang agak sulit di awal, tapi jika kita paham itu tidak terlalu sulit juga ternyata. Karena di sana udah ada audit trail, begitu auditor bikin appointment untuk audit, kita udah tinggal tidak terlalu susah lagi untuk melaksanakannya begitu. Data integrity mungkin pribadi bagi saya ibarat seorang anak yang tidak disukai oleh bapak kandungnya sendiri. Nah, mau atau nggak mau sebenarnya bapaknya juga harus menerima akhirnya. Karena ibu kandungnya adalah regulasi atau compliance yang harus dipenuhi di setiap perusahaan begitu. Jadi kalau untuk Amerika dan Eropa, jadi itu udah sangat umum di sana. Di Asia ini mau nggak mau juga akan mengikutinya begitu. Saya tergelitik melihat bagaimana kondisi farmasi secara global maupun secara nasional ya. Jadi di tahun 2020 berdasarkan revenue dari perusahaan besar grup farmasi di dunia, maka kita lihat posisi 1, 2, dan 3 itu misalnya Johnson & Johnson di 85 billion US, artinya sekitar 1.200 triliun kalau kita kali dengan kurs 15.000 rupiah. Di tempat kedua ada Roche ya, di 62 billion, artinya sekitar 1.000 triliun rupiah ya. Jadi revenue artinya over total penjualan mereka. Dan tempat 3 ada Novartis sekitar 700 triliun, diikuti oleh yang lainnya. Ini 20 besar global farmasi yang saya kutip dari Pharmaceutical Technology. Nah, kalau kita lihat top 20 farmasi Indonesia yang saya kutip dari Farmasi Industri, itu holding BUMN, Bio Pharma, Kimia Pharma, dan Indo Pharma yang gabungan 3 perusahaan itu menjadi nomor 1 karena digabung. Dan kalau kita melihat dari sebelum pandemi di Q3 2019 kita belum ada masuk COVID-19, itu pertumbuhan di sini masih positif. Bapak Ibu bisa lihat dari 1,5 hingga hampir 10% pertumbuhan di setiap perusahaan. Nah, setelah pandemi itu kita melihat pertumbuhan negatif minus. Dari yang plus itu hanya beberapa seperti Bayan Indonesia memproduksi vitamin, suplemen, AstraZeneca masih positif. Kemudian Soho kita tahu ada modulator. Nah, selain dari ini mungkin produk-produk ethical semuanya pasti drop. Tapi kalau kita bandingkan dengan industri yang lain seperti industri penerbangan maupun pariwisata yang pasti anjloknya luar biasa hingga 80 atau 90% minusnya. Tapi di farmasi ini masih bisa survive, mungkin masih minus sekitar secara total mungkin minus 5 sampai minus 10%. Dan masih ada yang pertumbuhan plus ya 22, 6%, 4%. Ya kita harap semua keadaan semakin membaik, dan di tahun ini kita sudah merasakan industri manufacturing sudah mulai bergerak ke arah positif. Ya. Kalau kita lihat ada perusahaan lokal ada 178 dan MNC ada 24. Jadi totalnya sekitar 200 company ditambah dengan company BUMN ya. Dan kita patut berbangga farmasi Indonesia itu di ASEAN itu terbesar. Memang kalau kita bandingkan dengan jumlah penduduk juga kita yang paling besar ya. Tapi memang perusahaan farmasi di Indonesia itu yang paling besar di ASEAN. Jadi melalui kita kembali ke Sotax Service, di mana mungkin Bapak Ibu sudah hafal ada IQ maupun MQ untuk Dissolution khususnya. Kemudian kami juga menyiapkan PVT di setiap customer. Untuk SPS sendiri sebenarnya Sotax Pharma Service, di mana kita bisa mengecek visibility, analytical, dan method development untuk produk ya. Jadi kunci mereka adalah for expert by expert. Untuk kontak, Bapak Ibu bisa mencatat nomor HP saya dan email jika belum punya, dan silakan kalau mau masuk ke gaia.co.id, ini produk-produk kami. Nah, ini untuk konfirmasi nama sertifikat yang nanti akan kami emailkan e-sertifikat ke email Bapak Ibu semua. Terutama ada gelar-gelar yang harus kami cantumkan, jadi tolong di WA ke saya ya Bapak Ibu ya. Baik, sampai di sini kita akan langsung menginvite Jurgen Kem. Jadi biasa kami panggil Jurgen. Jurgen adalah sales manager untuk kawasan Asia Pasifik.
Yes.
Yeah, Jurgen, now is your turn. You can start your presentation.
Thank you very much, Simon. I hope you can see the first slide.
Yes.
Yes, very good. Thank you very much and welcome everybody to this Lab Talk event. We definitely really appreciate this chance and position here to talk to every one of you who is attending and taking the time this afternoon. For me, it's just the morning because I'm sitting here in my office in Switzerland. So I'm just in between my coffee break and lunch break to talk to you. So we're very happy to present and share some of our solutions for automation and the aspect of data integrity with you all here. The presentation that I'm talking about is the different segments of automation and the linked data integrity that we have. Of course, there will be some overlap of what Simon was already talking about, but I will just touch the aspects on the automation parts on this one. And as an overview and to start into that presentation and company, I will just give you a quick overview over our company profile and the presence that we have on our global scale, followed then by the automation solution and the data aspects that we're focusing today. So basically, what we do have is three major hardware product segments. One is the product for dissolution testing, automated sample preparation, physical testing as well, all embedded into data management and customer services. I don't want to spend too much time on this one. Thanks, Simon. You already introduced this into the audience here. We are here also since more than 40 years in these field for pharmaceutical support and testing. Just as a few milestones, what we do have to talk about, of course, it is mainly, mainly the focus on the automated instruments that we are having from the USB4 apparatus that was built very early, but 91 introduced into the USB, but this is just one of the facts I will talk about later, just a little bit. Over the first fully automated compact dissolution system introduced in 93 over to the fully automated solutions that we have today. Foundations of companies and additions of other companies have also happened on the path of Sotax from the beginning to now. So we are a constant growing and expanding company and being a leader leading global player in the field of automation testing. So where can you find us? Of course, we are talking about Indonesia here, and you know that Gaia is our exclusive partner, but we also do have a global presence with direct and indirect partners all over the place, as you can see. So the focus, of course, for us and you here is around Indonesia and Asia. Also, the fact is that most companies propagate the importance of customer services, but only if you live up to this claim. So we do have a real worldwide network of partners, as well as very long time and exclusive distributors, as you already know, and like Gaia is our long-time partner in Indonesia. We do provide complete solutions for all of our customers. So as we know, we have these three segments of instruments showing. We also have the span of our products reaching from R&D in product development for dissolution sample preparation over to production testing, including IPC for tabletting machines and quality controls, especially for automated or fully automated solutions. So let's have a quick look onto the dissolution part and the automation that we are focusing on. The dissolution types and apparatuses 1, 2 as the major ones for automation, but also 5 and 6 are the functionalities that we provide. And as I mentioned earlier before, also USP4 is one of the topics. If you look on the USP 1, 2, 5, 6, there is, of course, a range, and thanks to Simon, I do not have to spend too much time onto these individual slides. But the key point is that we have a platform that is spanning from a manual system where you can just work individually on, over to systems that start with a certain amount of automation, all the way up to fully automated systems that work completely unattended for multiple testings. The way of data integrity is growing the further you move on from the automation level to the fully level. And of course, we have all these aspects covered by these systems. When I talk about automation, I will, of course, leave the manual approach on the side for this time, because there is everything to be done individually. Automation starts when the analyst is no longer fully involved in executing a test and data is collected independently. Semi-automation, as we call it, since it is for one dissolution run only, and preparation at the begin and end is still required. But the green part, as you see in that circle, is where automation is happening. And steps like starting and stopping is automation or integrated into the automation. We are collecting samples like into vials or into readers and being unattended in here. Configurations that are possible for those systems are seen here. So as a semi-automated system, as we call it, an offline system means you start to run, the system is collecting samples either for a single run or ideally for extended or delayed release in multiple samples during a profile dissolution run in tubes or vials, or we can integrate a UV for an UV online system to read samples directly, or having a combination of both. That means on and offline. So you could read samples and collect them as a backup, especially if this would be some kind of multi-component product where you have different finishing procedures, like one part could be done in UV, another part has to go to another UV that is not integrated in here, or into an LC system. Or we connect an LC system directly onto one of those systems, collecting and then transferring to an LC for a direct LC analysis. The same is available for double systems. A double system is a system where we just connect two of these bath systems that can have 6 to 8 vessels into one fully automated system. So we're running 2, that means 12 samples in parallel, which will be 12 samples collected in parallel or even analyzed into this one. So we have the same configuration types that are available on the manual and the semi-automated in the single way as we have it here on our double systems. All of those systems, of course, are running and controlled by a 21 CFR compliant software, which of course also provides the level of data integrity that is required to run these. Finally, what we are talking about and what is the highest focus is that only when multiple unattended runs can be executed in one sequence. This is what we call the fully automated approach, where not every instrument provider is reaching for. What you see here is a unique situation. So we at Sotax are the only company providing 3 different, but in the dissolution approach, equivalent fully automated dissolution systems. There's the 87 that has been introduced as the hollow shaft principle and hemispheric valve floor standing system for Sotax in the late 90s as the very first system, but still is available and still used with hundreds of systems across the globe. We do have the ATMD system, which is a modular and a very flexible benchtop system. So this is based on the latest model of the bath that you can see, this round design bath, which connects to multiple different modules to build your own system based on your needs for USP 1, USP 2, offline collection or online approaches. And we have a system that is called the ATF, which is a combination of the 2 systems, but still a floor standing system, and is the latest development in our portfolio. So currently we can provide any system that a customer just requires for automation just out from our pockets. How can we configure those systems? Again, the similar or same approach as we have for those semi-automated systems is possible here, of course, because we need to be able to scale up. So fully unattended runs from the preparation of a media over the dispensing into the vessel, checking the temperature, starting the dissolution run, taking samples, and at the end, doing a cleanup and prepare for a next run. This is what the fully automated systems are doing, and up to 10 batches in one sequence. So we can have this in a setup for offline, that means collecting in tubes or vials, or running a UV online, measuring directly into a UV cell, or having a combination of both as we have UV on offline, because in a sequence, you might have samples that might be for collection and samples that might be for direct online measuring. That also implements the idea that you can think of not running just one type of product. It could be multiple different products, since the systems can also switch between media. Here I'm running a short video for you, and I hope this is running properly in all the areas, just to show how these fully automated systems are working. So this was the overview over the fully automated dissolution systems as the using on the example on the ATF system. The way how the other two systems are working is exactly the similar approach, because for using fully automated dissolution, of course, we need to make sure that data is equivalent across our instrumentations. Having just a word about the USP4, because also here there is some automation possible. So of course, from the first drawing in the late 50s, as you can see this in this picture, to the first commercial instrument, it was quite a bit of a way. However, it was not until the 70s that the first conceptual drawings for a true apparatus received from the now creator of the flow-through method, that was Dr. Langenbucher. It's a very famous story with Sotax at the Swiss company called Gaia was manufactured. At that time, Sotax was a small engineering company, but now we are really considered as one of the global leaders in pharmaceutical testing solutions. So it has been pioneering since then in flow-through cell technology, designing the first prototype for that gentleman back in the 70s. Today, Sotax, of course, is still and will be considered as first in class and known throughout the world, with hundreds of companies using these apparatus. So all these instruments. The flow-through dissolution method really does offer complete flexibility on media volumes and allows repeatable positioning of virtually all kinds of dosage forms, such as powders, APIs, lipophilic forms, suppositories, suspensions, liposomes, microspheres, semi-solid implants, and medical devices, including drug-eluting stents. All this is described, of course, in the USP as the apparatus 4. The European Pharmacopoeia calls it a flow-through cell. Japanese is talking about apparatus 3, the Chinese as method 6, and of course, other pharmacopoeias. Looking into the type of automation that we can do here is very quite similar as the semi-automated approach. This system, of course, does not provide multiple runs or products that can be started, but it is kind of a semi-automation. So you prepare the system, start it, and then, of course, run it for the time that is required to execute that USP4 dissolution run, either in an open loop system, and that means we're collecting directly, in a closed loop where we're just circulating everything and probably reading this, or definitely reading this on a UV, or in a combination of both. So in a second approach, the whole setup could also be in a closed loop. Open loop means we're getting fresh media. Closed loop means we're having a real option of using the same media through a whole run of dissolution that is happening in here. All of these systems, of course, are controlled and driven by a 21 CFR compliant software and a database providing data integrity. Automated sample preparation. So this is also a segment where we are really pioneering in this area, and how we are doing this for the pharmaceutical industry since more than 25 years. Sample preparation is basically the task that has to be done for like content uniformity, assay testing, related substances, and impurity testing. All those tests that still have to be done, and all of you do in this, besides dissolution, and also other testings that has to be applied to your products. This has to be done we're doing this on a very compact and small footprint platform that is able to fast and very reliable extract a sample by transferring and applying most likely the same type of method approach as in the most manual methods. So extracting in a certain amount of media in a combination of organic, non-organic media like buffers, or finishing with diluting and sampling or directly injecting into LC systems, which is the typical finish nowadays for the most of those methods. Why do you want to automate this? And this is the task for any type of automation. The key is, of course, to prevent OOX. I'm not talking about only out of specification, there's also out of expectation and out of criteria. So this is key functionality that has to be avoided as much as possible, because this will increase the efficiency of your labs when you can reach a very high level of first right time results. So the types we do with the sample preparation platforms, there's two different platforms in work in a little bit different volumes. One is working up to 10 mils. That is the upper picture on that APW platform. The lower picture, that TPW that shows here, is a platform where we can do extractions from 10 over to a system that gives us the option to do up to 520 milliliters to use as an extraction volume. All of that connecting with an interface towards Empower. We're currently developing also another interface into Chromeleon. So this depends on what you're using currently, but it gives the option that a preparation can be unattended, transferred from dropping a tablet all the way to injecting into an LC without the need for transferring any data in this process. Also here, I would like to show you a short video on how these systems are running, how these systems are operating in real life. Extractions can be done with solid, semi-solids, and of course also liquid, as you have seen it, from CU over assays or swab extraction for cleaning validation. Systems have integrated balancers for recording sample weights, for recording correct volume dispensers, and making very high precise dilutions later on. Blend uniformity transfers and a very high efficient extraction of the samples with a high shear mixer. Filtering or for very difficult to clarify substances, an integrated centrifuge can also be applied to the system. Dilutions for a dilution rate from up to 10,000 from one sample can be applied in the system. Then either transferred into a vial for dissolution or injecting into an LC system that is connected to the platform directly, with the transfer of data to have an uninterrupted flow of data and integrity. Cleanup has to be provided. Here we have the same approach as in a dissolution system. So we have multiple samples. It could also be different samples into one sequence. So after the preparation, of course, a cleanup has to follow to make sure there is no carryover in between samples. Extractions of APIs on one platform can be done also with ultrasonic. And multiple batches of samples can be queued into one sequence. So let's head over to product range number 3, which is the physical testing and what we do see as automation over here. This is the overview picture and the summary of all tasks that you see on this slide. So from hardness over to PAT and IR. Of course, hardness means also thickness, width, diameter, weight, and break force if it is required. So this is already 5 parameters. It is more than just here seen on the picture. And all the other test classes are also able to get implemented into an automated and controlled environment. No need to run every test on an individual test sheet or different type of documentations. All these can be integrated into automation. I'm not spending too much time here. You have seen all these products already in the presentation of Simon. Thanks for that, Simon. So this is just a range for the physical testing, but we also do have many more. So third-party instruments that can be integrated into our environment that we'll show in a second. So here we have the development cycle of the physical testing for the hardness approach from a very first basic instrument, everything. And you see there's some kind of common thing. Everything goes back to the early 70s, where the first basic instrument was developed. And now we have in 2021, the state-of-the-art and industry reference fully automated station 8050 for lab and also for production. Also here, I would like to show you a little bit of an overview on how these instruments are flowing before then I'm heading over to the data integrity aspect for all the automation. Test classes, weight, diameter, length, different types of tablets. System has a patented way to separate and individualize tablets. Integrated balances. Tests are running in parallel on the same product. Making sure it is always and every time measured in the same direction. multiple batches can be set into a sequence run. So it is not only one type of product that can be started in a fully automated system, new AT4. So now let's head over to the topic of data management. The idea is to manage your workflow. So question also always comes up, where is data occurring? Literally at each and every step during the testing of the API in early R&D over to the manufacturing of the products. All related tests from R&D and QC and Q to QA for the final release of the batch towards the market for the patient. Better all data is safely recorded and available for any review or check. Of course, that's the great idea. The ideal solution is a single place for all data without any individual or just a minimized amount of records on paper or other places. No deviations, alterations, or potential fraud of data should be possible. From here, the data should be directly able to get transferred into a LIMS, like a laboratory information management system or an MES, manufacturing execution system, to merge with other related data for the individual product. Here's a little bit of a workflow for the data management software, from the administration and setup over to the operational tasks that are happening, from data acquisition, all the tasks that have to be done here, creating data, making decisions, making judgments, handling the data, exporting them, and analyze, of course, all the way to the release. This is leading to the fact that you need to have a very strong and integer system of data integrity. So we do have a platform that we call QDoc that has all this available. From manual to fully automated system, it is possible to keep track what did happen, when and by whom. So all relevant data points that are occurring, of course, will be documented and stored also in a secure database. The data management software should also be able to manage more than just one workstation, connecting multiple instruments onto different controllers with one software client in a networked environment, like we can see this here. Simon already had this screen in his presentation. Yeah? So why not having multiple different systems in a controlled and combined network, and we connect individual systems, modules onto one centralized database. All created data, all methods, all users with all the individual accesses and approvals are stored and handled in there. So there is one admin that is controlling all that systems, from dissolution over all the physical testings that we are having or that you are having in all of your instruments and laboratories. From running the instruments and doing all the executions, of course, the admin part is very significant. So reporting, this can be done at the place where the data is occurring and where the data is executed, but still very often this is not the case. QA is not located in the same building or in the same laboratories. So additional admin workstations can be still connected to that system, given access or specified access for what this task of the controller just should be. How's the data integrity thing looking in here? So data integrity was and still is, of course, a hot topic in the pharmaceutical industry throughout the last 15 years or more, mainly since the FDA and other authorities started looking very closely into this. Largely where it were years in the beginning where non-compliances were headlines. We all saw this in the news in the pharmaceutical world. Regulators were probably turned away by drug companies. Contract research organizations faced serious charges of data manipulations. Warning letters and import alerts were issued due to data integrity concerns. Here you can see just a graph where this was just moving and going up from 2008, where yes, we should do something over the way. We should do something where really now auditors were really looking into specifically. This was in 2014, 15, and all of a sudden there was a huge increase of that because companies were not ready for data integrity. Interestingly is that non-compliances weren't only limited to India and China. That was always the big areas where this was found. But these years also saw lots of cross-border activities of European regulators across issuing compliance notifications to American companies, while the FDA was more heading over into Europe. So there was a crossover control from Europe to Americas. And also data integrity issues were triggered more than 1/3 of all regulatory actions since 2016. So this is very, very much focused in here. The question that with data integrity is happening is the mystery that, oh, where's my dissolution data? It happened? No, it disappeared. We don't know what happened. So of course, everyone knows that letter of the FDA that is talking about a fully controlled audit trail should be integrated and expected that facilities should upgrade to an audit trail by the end of 2017. This is based on that audit trail letter of the FDA that was existing already years ago. So we already passed 2017, but we still see that lots of those tasks were still not integrated. Yeah. So, and this is the question of how is that possible with being compliant, but also having the factor of costs in mind. Data integrity that definitely includes 21 CFR Part 11 compliance is, of course, important, but most companies but really looking just in 21 CFR and to have this as a fix for CAPAs that they have to fulfill. But this is the beginning, but surely not the final solution. Part 11 is talking about the electronic data only. All data that we have seen in this data stream, of course, is metadata. And this is all leading to the term, how do we deal with the fact of data governance? That means the handling of all data that begin from the end and to beginning from to the end of the product cycle. So 21 CFR 11 is just a component of the whole integrity. Although, I have to admit, a very important one, since we as a solution provider, of course, have to and do meet the criteria to get accepted by our customers and the authorities as well by providing systems that are compliant to this. So there is all these aspects from authentication, storage, master data, audit trail being valid for the intended purpose. Of course, the compliance approach is to avoid to break processes, not to do too many or getting away, of course, ideally from manual data recordings that could be altered, that could be lost, like the magical, where's my data going? We don't know. We all do know the terms about ALCOA, and this is very important to keep in mind. So it is stressing, but we keep stressing this fact that data and the fact of ALCOA is for attributable. That means who is created record and when, because it should be also important to say who amended the record when and why. Legibly, that is, basically it should be common sense. When you write something down, it is not just about you and your colleagues are able to be able reading this record, because an auditor is probably not being able to read the handwriting of everyone. So we're not like the doctors and pharmacists that sign the receipts and the recipes that you have for your prescriptions. So we do make sure everything is identifiable by anyone. Contemporaneously means also it has to happen at the same time, the record when the test was executed, not like, okay, I'm doing my test now and in the afternoon, oh, damn, I have forgotten to do some recordings. I will do this later then. Original data, no manual transcription. That means create the data and save them directly. And of course, make sure data is accurate. That means correctly measured. That implements that if you have devices that you take for measuring, make sure those devices are calibrated and adjusted to the proper need. There is a focus also on a term on Alcoa Plus. So having all these Alcoa principles applied, it also is important to make sure that in fully automated systems that provide data integrity, that record data, that there's also a completeness of all the data. So I need to make sure the data is not just existing, they also need to be complete and available. They need to be consistent. That means I have my entries in a chronological order, and they need to be accessible over time. This is where IT companies or IT departments, of course, come into place at the customer side to make sure the data can be transferred over from time to time. The data integrity principles of this Alcoa and Alcoa Plus here in a circle is seen from a pharmaceutical laboratory point of view, where data management and governance is important with different pillars like the organizational structure. That means what is happening, what are we doing, the operations, who is doing something, and the utilities, how are we doing this and with what. Methods and assays are regulated in all of those examples of guidances that I'm outlining here. That is something that you definitely have to do on your daily business, but it is important to follow those. Materials and reagents, that means activities from receipts till approval. Make sure you have all your required materials and samples available and in place when you need them. Control and document whatever you do. Facilities, equipments, and the computerized system management means have your instruments in a constant and high qualification status. We cannot stress this much enough because this is really key. An instrument that is out of calibration, an instrument that is not working properly is definitely giving you troublesome data in the end, and an investigation could be quite cumbersome in here. And for me, one of the most important parts to have this is, of course, the fact of the personnel and training. Keep your staff on a constant, really high and educated level. Make sure everyone is aware of changes, making sure you know that people know their SOPs exist, have key users for instruments kept. As a personal experience I see with tons of my customers is using automation is not just a simple task. It requires highly qualified people. You want to qualify people and make sure these people do use the systems properly. Development, your staff, promotions, that's all the parts that you do anyway. And of course, for some of you, and this is something that Jonah will talk about later, the challenge of outsourcing, finding the right partners in the market. If you have to reduce costs, if you need to use standardized or controlled equipment, either I'm outsourcing to somebody else and make sure they're using my standards, or I'm getting the one who is the outsourcing company. So I need to apply to those standards that my partner is asking for, as well as being able and appropriate to deliver in time. All those things, of course, and I don't want to stress this too much now, in terms of data integrity, there's really tons of material that everyone can find on here. What I want to close this presentation is, of course, that we committed to be 100% customer focused. So the automation with the use of data integrity is really and definitely keeping costs under control because you can plan. It's keeping also your workflows under a well and very good control. And also it keeps your metadata or the data under control because you keep them in a safe place. That means your processes are well under control. The advantage of an automated system is you don't have variation in how something is executed, which is a very, very important topic here in this discussion, as well as keeping the products more safe. all the end, this leads to the fact that if everything is aware and available, this keeps also your patients more safe and keeps your business up and running. So here I'm liking to finish my presentation, and we are open to the Q&A session. So thank you very much for your attention. I hope I could shine a little bit of light into the automation world of Sotax and the aspects of data integrity for you here.
Okay, thank you, Jürgen. Now the Q&A box.
Yeah.
We have 3 questions, 2 for you and 1 for me. Okay. Can you see the?
Yes, I just opened it up. So how to avoid or minimize the deviations from one instrument to the other instrument's testing result, or from one equipment brand to another brand's. So if you are transferring a method, so especially let's say this is something that is typically happening in the dissolution part where we have this. You have one brand of dissolution, you have one instrument of dissolution, and you want to scale up either to a semi-automated or a fully automated system. Basically, the dissolution makes this pretty much easy because the approach or the method is described. So you know exactly, you do have the same media, you do have the same amount of media, the apparatus, temperature, timings, this should be the same. And also the design of the vessels should be very similar. So deviations, so when you do a transfer, you definitely have to do a certain transfer validation if you're transferring from one apparatus or from one instrument to the other. So you cannot just say, okay, today I'm using blue, tomorrow I'm using yellow. So you have to make sure that the result from yellow is equivalent with blue. And equivalent is the key word here. So you have to apply a validation approach where you do testing on the blue instrument or on the yellow instrument, saying, okay, this is what I get here. You transfer and run this on your other instrument, and you have to assign this within equivalency range. Typically, depending on products and methods, this is something within 2 to X% that you need to see equivalency, and you validate this. So you validate one batch here and run it, say, this is my result. You run this on the other instrument and compare and define the equivalency. This is how you would also prove to authorities that you have done an transfer from one instrument to another. So this is something that you can do with support of us, or even we can outsource this into other companies like SPS that I've mentioned. I hope this explains this. So if there's more questions, we can answer this a little bit later then as well. Simon, do you want to speak to the second one? Because then I can look into question 3.
Yes. The question to me, Oke, apakah yang dilakukan untuk mempersaingkan brand supply line terkait service di luar Jawa? Nah, ini terkait dengan customer. Memang kalau kita lihat perusahaan farmasi itu sekitar 60-70% ada di kawasan Jabodetabek dan Bandung dan sekitarnya. Nah, mungkin 20-30% ada di Jawa Tengah dan Jawa Timur, dan ada 5% yang di luar Jawa, mungkin kayak saya. Nah, ini untuk yang selama ini yang kami lakukan untuk customer, jika memang perusahaan atau Bapak Ibu membutuhkan support yang sangat spesial dari kami menawarkan jasa service kontrak. Jadi setelah melewati masa garansi 1 tahun atau 2 tahun biasanya kami berikan, itu ada service kontrak untuk alat tersebut, sehingga kami bisa melakukan visit berkala, misalnya setiap minimal 6 bulan sekali untuk visit untuk maintenance, preventive maintenance alat tersebut, begitu Pak Zainal. Untuk tambahan dari jawaban Jürgen untuk yang deviasi.
Okay, my task, Simon.
Sorry. this is additional answer for the first question from Pak Zainal. Jadi Pak Zainal, untuk perbedaan antar alat maupun antar brand itu bisa saja terjadi karena semuanya sama-sama memenuhi USP. Misalnya kita bilang aja dissolution tester. Jadi antara merek Sotax dengan merek lain itu bisa terjadi perbedaan hasil. Misalnya di bawah 3% kah, atau bisa jadi 5%. Itu kenapa? Karena USP sendiri misalnya kan memberikan suatu range. Misalnya diameter dari vessel adalah 96 sampai 104 mm diameternya. Nah, misalnya Sotax itu 98. Nah, kalau kita lihat misalnya merek yang lain ada yang 103 atau 102. Nah, dari ukuran vessel bisa berbeda. Jadi sehingga kelarutan bisa juga berbeda. Sotax lebih tinggi atau mungkin merek lain lebih tinggi, itu bisa terjadi. Jadi untuk meminimas memang agak sulit ya Pak Zainal ya. Jadi yang penting tadi kata Jurgen untuk bagaimana mengekivalenkan sehingga meminimize perbedaan antar alat maupun antar brand begitu. Seperti misalnya di hardness tester, ada yang menggunakan constant speed maupun constant force. Nah, itu perbedaannya bisa sekitar 2-3% itu udah pernah kami lakukan juga. Disintegration tester juga begitu. Ada yang pakai magnetic, Sotax pakai discontact. Nah, itu perbedaannya bisa yang magnetic lebih lambat, yang discontact bisa lebih cepat begitu. Itu bisa selisihnya signifikan juga. Misalnya endpoint-nya itu 10 menit, harusnya Sotax itu sekitar 9 menit udah kontak, yang merek yang lain bisa sampai 12-13 menit begitu. Di mana pengujian manual yang asli itu sekitar 10 menit seperti itu. Nah, oke, itu saja mungkin tambahannya. For the next question is for you, Jurgen, you can.
Yes, I already typed an answer. So that was the question about surfactants. Of course, if you use a surfactant, you can still use this within a fully automated system. So surfactants, you definitely not do degas or deaerate. So you can still use a surfactant with a concentration up to 2 to 3% easily, depending on what type of surfactant you're using. I've seen also a little bit higher, but typically it is in a range up to 3%. That could be used also as a media in the fully automated systems. The system can still dispense. The foaming is not that critical. It only happens when you would degas. So usually the degas is not happening with the surfactant. It can be easily handled. Do you need to translate this, Simon, or is that okay?
Just additionally for the question, I think for surfactant it depends on the how to sampling with the manual or auto sampling. If we're talking about the auto sampling, it means if you use the searing pump, the surfactant will be issued because of some bubble will stuck in the searing. So we have many experience in customer using other brand that they use searing pump cannot solve the problem. So finally they choose the piston pump.
Yes.
Sotax, so it solves the problem.
Exactly. So yeah. Syringes are not the choice for surfactant media. So a piston pump, which is in all of our the automated systems, are not using those syringe pumps. So the ones that I was talking and showing about were all piston pumps, and these piston pumps have absolutely no issue with surfactant medias. Okay, so there's another question. If the data of this solution is fully automatically, our LIMS can be connected with that instrument. So that data package called QDoc that I was introducing can connect to a LIMS system. So it can be applied that an automatic transfer is there. Since every LIMS system provider, of course, has different ways on connecting or a communication protocol, we can create an individual, let's say, link to every LIMS system. So if this is a requirement, we can collect all the data and we can create a handshake or an interface to any kind of LIMS system with the customer. Any kind means, of course, the most common. We need to talk to the IT groups, but those data can straight be, after the collection, either be transferred over, or if a customer decides to do the judgment first within the QDoc and then doing the transfer. So there's vice versa possible. The answer is yes, data can be automatically transferred into a LIMS system. So another question coming from Ivan is, how about calibration of the automation sample prep? Is that gravimetric calibration? Yes. How about the volume dilution is bigger? Is that optional part equal with over 250 volumetric flask and sonication system? Is there in sample preparation? That's a few questions already typed in there. So how is that automated sample preparation working? So it's based on gravimetric confirmations, either from volumes and also weights. That means balances are integrated and those are calibrated. Yeah, so it's a standardized analytical balance that are integrated in that system. You can usually calibrate them and check them as you would use your regular, analytical balances that you are using in the laboratories. So the calibration factor is the same. The volume that is required, so the range of extraction that we can go with the sample preparation is up to 520 milliliters. So this is the maximum. The range goes from an extraction vessel size 20 to 520. So if it is more than 250, that is not a problem. I have done lots of methods that was using more. And of course, it is always a question of the solubility of your sample. So the extraction is not an issue. The way how it is, the sonication system in the sample preparation, this is only for the platform called APW. More details you can definitely get from Simon. Please contact our partner Gaia. They can provide you with more individual details, and I'm here to answer more questions in terms of applications. But as I was seeing, there were two major platforms, one with the extraction with a vessel which had a homogenizer, and the other platform is using an ultrasonic probe to extract samples into a tube. So what is the existing manufacturer that implemented sample preparation? I didn't talk about this. Yes, we do have these platforms already since also more than 20 years, over to the 5th generation where we are. We have sold hundreds, 500s of those platforms into the pharmaceutical industry since then. Currently, we still have more than 250 active instruments in industry and in the pharmaceutical world globally, from major companies, innovators, over to the small generic companies. Major companies like AstraZeneca, GSK, Bayer, all those companies are using somewhere in the world these platforms, over to small companies that they just use them locally. So we have a global presence with all of those solutions that I was showing in my presentation, with thousands of instruments currently installed.
Okay, thank you very much for the answer, Jürgen. Dan terima kasih banyak untuk pertanyaan Bapak Ibu, Bapak Jainal, Ibu Meyar, Pak Andung, dan Pak Ivan. Kita tidak ada feedback dari answer, jadi mohon maaf, kami harus langsung lompat ke pembicara berikutnya. The second speaker is from Song Louis from SPS Pharma Service. Jadi kami udah bekerja sama dengan John Louis sekitar 20 tahun ya. Dan beliau sebenarnya yang merintis Sotax Asia, dan saat ini diteruskan oleh Jürgen, dan sekarang John Louis sendiri fokus di SPS Business Development Manager. Jadi turun gunung nih ceritanya. Ya, for the time, please John Louis for the presentation. It's your turn.
Thank you, Simon. Thank you. I will share my screen. Do you see my screen? Yes, I guess. So welcome to all of you. Good afternoon. And I will say in Indonesian, I know some words, just selamat datang, selamat datang. I hope my accent is understandable. Thank you very much for participating, attending this webinar. I'm extremely happy today to present you the company SPS. We heard a lot from Jürgen about system automation, data integrity. Now let's talk more about application, in fact. So who is SPS? In fact, is CRO, it's a contract research organization. And we used to say this CRO is specialized in complex dosage form testing. And I will explain to you in details. So that will be the agenda of the presentation. Of course, we start by who we are, expertise, and developing a little bit the services of SPS Pharma. So who we are, in fact, we created SPS 15 years ago. We are in France, we are not far from Paris, and it's a CRO who is offering, of course, all analytical services. But what makes difference between other CROs is really SPS is the only one company in the world specialized in R&D for disso and release testing. It's an important point because it's really rare to have a company only specialized in this field. And I make the link with what Jürgen was saying because we talk a lot about automation. A lot of question comes to us in terms of automation validation, transfer method, and this kind of aspect. And I will, of course, detail that with you later on. Another very important aspect is this company and this lab is full GMP compliant facility. Also, it is many times US FDA inspected, so GMP and FDA inspected. And of course, we have regularly audit of pharma company, client of SPS visiting us and of course, developing the audit with us. Client base, as you can see, we are based in France and based in Europe, but only 40% of the clients are from Europe. Most of them are from America or also from Asia. So as a summary, I repeat, we are FDA, of course, inspected, CGMP lab. We have multiple equipment inside. I was hearing Jürgen answering an important question of comparison between dissolution brand to another dissolution brand, or even from manual to automatic way. Of course, SPS is doing that regularly in our lab, and we are just doing this kind of test really to prove that, of course, from manual to automation, we can move easily, smoothly, or from one brand to another brand, we can avoid discrepancy. That was an important question that you're going to answer as well. We, of course, have regular interaction with industry and health agency, and we also work not only for human, but also for vet, for animal health. We have the questions on them. In terms of organization, we are really, I would say it's a flat organization. I'm in charge of the sales and business development, and as you are organized in your pharma, we have a head of lab. In fact, we have two. We have QC and, of course, R&D as well, with every project leader running their own project. We also have a pharmacist, which is a QA person, qualified person, in fact. I will not go in details because that you know that very well. Of course, we have a full GMP quality system in place, including, of course, personnel, facilities, documentation, equipment, lab operation, and of course, the full quality management, where you have some deviation management and so on and so on. But you know this aspect very well. It's just a reminder. So our expertise, let's take a look at our expertise. In fact, I like this slide because it shows, in fact, where the dissolution aspect interact, in fact. We start with the APIs, we go to the formulation, we go to analytical, of course, clinical, and until the production. At every aspect of the pharma steps, in fact, the dissolution play a very important role. We have many times clients coming to SPS with differences in batch to batch, not understanding why there are some differences, because of course production didn't change, nothing, but for example, they change their APIs, and they, instead of purchasing their APIs in Italy, now they purchase them in China or in India, same APIs, but of course, giving different profile for the finished product. So it's important also that to understand that we totally understand at SPS this interdisciplinary concept. And we face challenges of your client. You come with challenges, and most of the time, people don't even know if USP1 or USP2 is the right method. Why not USP3, 4, 5, 6, or 7? You know, we have 7 USP methods described in a pharmacopoeia. So of course, SPS is equipped with different equipment from 1 to 7. And what makes difference as well, it's a know-how that we have. If you come with stents, if you come with emulsion, if you come with suspension, or just sustain release product, we can really guide you, because we get used to work on this issue. And we do also a lot of IV, IVC at the R&D level. I will explain to you that later on. So let's take a look briefly at our type of service that we provide. So we have 3 types, in fact. Let's start by R&D services. I just explained you the difference between APIs sometimes. So we do API characterization, full API characterization, using intrinsic dissolution, also using extrinsic, like USP4 system. We do more, we do microscopy and so on, to really analyze and to understand the differences between your first APIs and second APIs. Who knows what happened? We also do analytical method development. So we have UV, we have HPLC, of course we have UPLC as well. We had a question last time, do we do only that link to dissolution? No, of course, if you need help in developing your analytical method, we can do it for you, not automatically link to dissolution. One of the key words, of course, is feasibility study. Feasibility studies, you come with a form and you really don't know which technique to use, and it's not so easy. You cannot buy 7 machines for sure. It's impossible. Maybe you have some in your lab. Maybe it's complex to have it. If you have a USP4 system in your lab, which configuration? Closed system, open system, of course. We were counting, we have 12 USP4 systems in the lab of SPS running simultaneously. So we are fully equipped in order for us to do the feasibility study for you. Having done the feasibility study, of course, we can do the step 2, which is the dissolution method development. And of course, step by step, we test all the aspects of the method development. For example, the flow rate, the cell setup for USP4, the filter as well. I was hearing the question about surfactant as well. Impact of surfactant, we try to avoid, but we cannot use organics, so we minimize this kind of thing. So we really develop the method for you. An important aspect, and totally linked to what Jürgen was explaining to you, method automation. So what do we do? It's a method automation at SPS. For example, you are really interested in knowing more about the concept of dissolution automation or content uniformity automation, sample prep automation. For sure, we can do demonstration. A demonstration is not a method development. It's not even a feasibility testing because it takes more time, you see. So we have a lot of requests from clients. We are about to think about automation. Say, okay, maybe this automation concept can help me to improve my process or to be more, I would say, proactive and more productive in my lab. So the first step is really to analyze, to evaluate maybe 2, 3, 4 products maximum, to send that to SPS. And of course, SPS will do what we call feasibility studies. We try to minimize the steps and to really see if, first of all, it's possible to automate. Most of the time it is possible. And of course, to develop for you the first method parameter. And it gives you right away an idea if, yes, you can pursue in this automation concept, or no, it's not so interesting for you. But again, I repeat, most of the time it is interesting for you. You have just to choose the right product in order to, of course, automate the method based on that. So we do that at SPS. We help the user, in fact, who are thinking to automate in his lab by doing this feasibility testing. Having done the method development, of course, we do what you know well as well, the method validation, and most often also the revalidation. And the last step, of course, that we do the method transfer. We can even do method transfer physically to your lab. Some people from SPS can come to your lab and not only doing the training, but do transferring the method. I take an example of TPW that Jürgen was explaining just before in terms of sample prep. Most of the time when you use SPS upfront, as soon as the machine is delivered to your lab, we do the IQ/OQ training, and then of course SPS can come and do what we call application week. And of course, you can start to work day one on your given product. You save a lot of time, and Jürgen mentioned it. You save time, you save cost, in fact. That's about it for the R&D services. Now let's take a look at I would say the routine analytical services, the GMP part. Of course, as we are GMP and FDA approved, we do the QC. We do the QC analysis using all kind of technique for dissolution. We do the assay and degradation product. We also do UV, HPLC, UPLC testing. And as Simon mentioned as well, we have physical testing in our lab, and we do this kind of thing. So why outsourcing to SPS? Many reasons, and Jürgen mentioned it a little bit. We can say lack of space, for example, or lack of capital investment, or just a peak in your request, you know. We see that a lot because we do also stability studies. Most of the people now outsource the stability studies. It's kind of boring things to do, time consuming. You need space, of course, because you need this ICH climatic chamber. We do have all of that, and of course we do that for our client. So we do the full stability following the ICH guideline anywhere. Okay. Last step, of course, we do also, as we are GMP and FDA approved, we are like a pharmaceutical establishment. We do the clinical and commercial batch release for you, of course, because we have a qualified person signing the document. Last but not least, in terms of support services, and some question for Jürgen, we are really linking this kind of support servicing to what I will say now. We do troubleshooting, we do investigation. You can do it yourself, of course, it takes time sometimes. And maybe you have not so much experience with it and you don't understand why you have differences from one dissolution machine to another one, or one dissolution machine manual versus one automation one. So of course, we do this troubleshooting for you. We do investigation. We can help you to go again faster, fixing your issue. What we do a lot as well is consulting. Consulting, we go to your lab, or even without physically going, we exchange information. We review your analytical and clinical data from, for example, failed bioequivalence. A lot of cases now coming from China, for example, failed bioequivalence, and it's due to, in fact, the dissolution. I give you just an example. USP2 is a method of choice. In fact, they were doing a USP2 in routine testing, but finally they failed bioequivalence. So it just happened that, in fact, USP2 was not discriminative enough. So they came back to us and said, okay, could you find a dissolution method which is really discriminative and mimicking my bioequivalences? And we are using USP4 for this type of product. So of course, USP4 could show exactly the same differences from batch to batch, in vitro versus in vivo in bioequivalences. So of course, we do that as a consulting. We do help you if you are facing some OS situation, and of course, it can come also from the APIs. Don't even think about the process, but most of the case, we have to go down to the main root cause, which is probably the APIs. We do audit. We do, of course, testing contractor, and last but not least, we do training. So of course, we do a lot of training. People ask us to come to your lab maybe 1 day or 2 days, training the people how to work in dissolution testing, how to do, of course, the proper way dissolution testing, or the validation way, because we have a lot of questions about validation nowadays. And of course, we do a lot of IV, IVC. Of course, we do not do the in vivo part. You provide us the in vivo part. We do not do any clinical studies, but we do all the rest, of course, all the correlation and so on and so on. Keep in mind, we can do also testing on non-conventional dosage form, drug-eluting stents, implantable form, you name it. There is so many different forms and difficult forms on the market that we can do, of course, using the USP dissolution system, but also using non-conventional dissolution technique that we did implement in our lab. And of course, we have this possibility of GMP implementation to finish on that. So this slide is just to show you, and you know that very well as well, from again, the API to the commercial post-approval, where SPS can help you, in fact, from the APIs to the formulation, to the final dosage form, and of course, to the post-approval. At any steps, SPS can help you because the impact of dissolution is everywhere. So it's an important important slide to remember that SPS is not only dissolution method development. We really interact in every steps in your pharma process, and we can help you, of course, step by step in your process. I think that was my last slide. It was quite a quick overview of SPS Pharma Services. So thank you very much. And I'm open now to your question, any application question you will have. Of course, maybe not all application I can answer now, but please feel free to send me your question now. Thank you very much.
Bapak Ibu semua, kalau ada yang pertanyaan seputar SPS Pharma, silakan ditanyakan di Q&A box-nya. The last, Zhong-Louis.
Yeah.
The last of your presentation, I'm quite interested with your. You said that SPS provide non-conventional method. What it mean?
We sometimes for some application, Simon, we cannot use USP 1, 2, or 7. It's very specific. We have to increase some flow rate and so on and so on. So we develop some very specific machine in order to really do the in vitro release testing. Of course, it's a R&D level, not QC level. But yes, we did develop some specific approach for doing in vitro release, mimicking, in fact, what happened in vivo. That's the concept, yes. I'm going to give you an example. We use dialysis bag, for example, for a nanosphere, which is a difficult product, of course. We cannot filter so easily. So we use some dialysis bags that we insert inside the USP4 cells, and it's working absolutely fine. I would say USP4 is compoundial method, but the dialysis bag inside is not described anywhere. But slowly but surely, when we develop something and people start to work with it, it can become, of course, compoundial, of course. But the FDA accepts whatever you developed. If you can prove that you have no other choice to work this way, there's absolutely no issue to use this way of working. Nanosphere is a good example. We have multiple companies already working on Nanosphere using USP4 technology and using the dialysis bag inside the cell, even though it's not written or described anywhere.
Okay. So it is the development of the variation of the USP4, right? It's not but not official conventional
no,
method.
Exactly. USP4 is official, but inside USP4 you can put whatever you want.
Correct.
And we always try to find the best solution for our user, for our client.
Okay, good. I just thought that you are trying to search for the USB8.
Never. It will never happen. I can just say something about it because I was a USP4 expert committee member at Washington, and clearly when we were thinking about having a USP8, no way. There's a front cell for cream, for example, exists. It's not USB 8, but it exists as well. But we can do also cream on USB 4.
Okay.
So it will not be USB 8 anymore.
Yes, I think 7 method is just enough, right?
I think so. I think so. Validation is a key point.
Yes. I think no Q&A box is empty. So I hope all your presentation is clear for all the participants.
I hope so.
And yeah, we still have 5 minutes to finish. So Bapak Ibu kalau ada pertanyaan silakan masuk ke Q&A. Kalau tidak mungkin nanti sebenarnya untuk automation, data integrity dan SPL Pharma untuk presentasi lebih detail, Bapak Ibu bisa menginvite atau menginfokan ke kami. Misalnya Bapak Ibu butuh informasi lebih lanjut, bisa hubungin ke HP saya ataupun ke email, sehingga kami bisa memberikan sesi yang lebih spesial untuk perusahaan Bapak Ibu, untuk kami follow up online meeting presentation yang lebih detail begitu. Dan untuk apa namanya sertifikat nanti yang akan kami bagikan, sehingga nanti kami akan mengontak Bapak Ibu setelah kami dapat data dari Lab Indonesia, Ibu Dewi, sehingga nanti kami akan follow up untuk mengontak Ibu sekaligus mengirimkan e-sertifikat dan untuk top up e-walletnya untuk mengganti kuota yang Bapak Ibu habiskan selama webinar ini begitu. Tanda terima kasih kami pada Bapak Ibu. Ya, mungkin Bapak Ibu bisa mencatat nomor saya dan email agar nanti untuk verifikasi data nama Bapak Ibu yang akan dimasukkan ke sertifikatnya.
Baik, Pak Simon.
Ya, mungkin cukup ya.
Terima kasih sekali, Pak Simon. Sebelum kami mengakhiri webinar hari ini, kami mohon kesediaan seluruh peserta untuk dapat mengisi polling yang akan kami tampilkan pada layar Anda. Polling pertanyaannya adalah sebagai berikut. 1. Menurut Anda, apakah webinar ini memberikan informasi dan manfaat untuk Anda? Kedua, menurut Anda, apakah informasi yang disampaikan oleh pembicara sudah sesuai dengan tema webinar? Ketiga, jika kami menyelenggarakan webinar berikutnya, apakah Anda akan berpartisipasi kembali? Terima kasih kepada peserta yang telah mengisi polling. Kami informasikan kepada seluruh peserta, jangan lupa follow sosial media kami untuk mengetahui update terbaru dari industri lab di Lab Indonesia Instagram @lab_Indonesia, Lab Indonesia Facebook @lab_Indonesia, website di www.lab-indonesia.com, serta YouTube di Lab Indonesia. Serta follow juga PT Gaia Science Indonesia Instagram, Facebook, Twitter @GaiaScienceID, serta website di www.gaiascience.co.id. Dengan demikian, webinar hari ini telah usai. Terima kasih kepada moderator dan pembicara yang telah memberikan informasi pada webinar hari ini. Semoga bermanfaat bagi kita semua. Terima kasih juga kami ucapkan kepada seluruh peserta yang telah berpartisipasi. Sampai jumpa lagi di webinar mendatang. Stay safe and healthy.
Terima kasih semuanya. Salam sehat. Thank you, Son Louis. Thank you, Jugen. Makasih, Bu Dewi.
Thank you, Sam.
Terima kasih Pak Simon. Thank you Mr. Jean-Louis. Mr.
Thank you, everyone.
Jurgen, thank
Yeah, it
you.
was a pleasure.
Bu Dewi, untuk data partisipannya kami tunggu ya, Bu.
Thank you, Devi. Thank you, Christy. Thank you for everyone.
Thank you. Thank you. Iya Pak Simon, nanti akan kami kirimkan segera ya Pak.
Oh ya, baik, Bu. Makasih, Bu. Oke. Hah?
Good.
Jean-Louis,
Yeah.
Jugen.
Simon, see you soon in Indonesia.
Yes.
Jakarta.
Thank you for your help.
Yeah.
Thank you for the presentation.
Okay, we stay in contact. Thank
Very
you
good
very much.
presentation.
Thanks.
Bye
Stay
bye.
safe.
Bye-bye.
My best regards to your
Same to
family. Yeah, bye.
Bye-bye, Dewi.
Bye.
Bye.